Hello, and welcome to today's AAPS eChalkTalk, hosted by Certara. You may input your questions at any time using the questions bar of the GoToMeetings panel. It is my pleasure to introduce our moderator, Dr. Suzanne Minton.
All right, thank you so much, Stacy, and thank you so much to AAPS for hosting us today in this webinar. Today's presentation is Avoid the Valley of Death, Increase Confidence in Translational Research Using Biosimulation. On the next slide, I'll introduce our two esteemed speakers today. So first up, we have Dr. Mark Lovern, the session description, you can read it, and here's the objectives, and we'll get into this throughout the session. So next slide, please. Okay, so our speaker today is Dr. Mark Lovern. He's currently a senior vice president at Certara in the Simcyp group. Mark is an expert in modeling and simulation with more than 20 years of experience applying modeling and simulation approaches to drug development. He's taught numerous technical training courses and lectures and workshops. He's worked in both the pharmaceutical industry as well as consultancy companies, and he will be joined in the Q&A session by Dr. Hannah Jones. So next slide, please. Hannah is currently a senior vice president and head of our PBPK consultancy at the Simcyp group in Certara. She's spent more than 20 years in pharma. Mark, you've got, we somehow are out of the slideshow mode.
Are you looking at my presenter screen?
Yeah, okay, but you're back now, perfect. Okay, thank you. Hannah's spent more than 20 years in the industry, and she's published more than 60 publications on PBPK modeling and other DMPK related topics. She has considerable influence. I don't know why it keeps going back and forth. Influencing drug research and development programs through modeling and simulation. Okay, you're good now. Let's just hope it stays.
I swear I'm not changing anything, so I don't know why it's doing this.
Okay, it's all good. So welcome to Hannah and Mark, and now I'll hand it over to Mark to begin the presentation.
Thank you, Suzanne. Before delving into the scientific discussion, I would like to say a few words about the company that Hannah, Suzanne, and I all work for, Certara. Certara is the world leader in biosimulation and model-informed drug development. We have over 1,200 employees and 2,000 client companies worldwide. In the past 4 years, we have contributed to 250 plus regulatory and payer submissions. And in the last 8 years, over 90% of FDA submissions were supported by Certara software and services. Now, let's set the stage for our main presentation today. The phrase Valley of Death was coined in response to the severe attrition rates historically experienced during the preclinical phase of drug development. For example, only 10% of preclinical drug research projects eventually progress into human trials. This attrition is due at least in part to the inability to either reproduce experimental results or meaningfully translate such results to humans. The term biosimulation refers to the use of in silico models of biological systems to predict how the body affects the drug and how the drug affects the body. Biosimulation can not only help to reduce attrition rates during the preclinical phase of drug development, but also reduce the cost and time needed to attain IND approval. It allows for quick elimination of compounds that have no promise, streamlines preclinical and clinical studies, and helps determine first-in-human dosing. Because of its potential to enhance drug development decision-making and reduce costs, application of biosimulation is increasingly encouraged by global regulators. Simcyp is Certara's business unit specializing in mechanistic biosimulation. Generally, our capabilities fall under one of three categories. One, physiologically-based pharmacokinetics, or PBPK, in other words, what the body does to the drug. Or two, quantitative systems pharmacology, what you want the drug to do to the body. Or three, quantitative systems toxicology and safety, what you don't want the drug to do to the body. The focus of today's talk will be on PBPK. The Simcyp simulator, which was developed by Certara, is the pharmaceutical industry's most sophisticated PBPK platform. PBPK methodologies allow us to predict what happens to a drug once it is introduced into a human or animal. PBPK models generally describe the absorption of the drug into the body, its distribution to relevant organ systems via the circulatory system, and its eventual metabolism and excretion. PBPK model components usually have a direct physiologic interpretation, such as organs, tissues, blood flows, etc. This is in stark contrast to classical PK models, in which most compartments, aside from the central blood pool, are empirical constructs. PBPK models may be used to generate virtual individuals based on such characteristics as organ sizes, cardiac output, and enzymatic phenotype, among others. By simulating multiple individuals and incorporating between subject variability into the simulations, virtual populations may also be derived. PBPK simulations also incorporate characteristics specific to the drug or drugs of interest, such as physicochemical properties or phys-chem, as well as what transporters or enzymes are involved in the drug's distribution and metabolism. Such data may be derived from either in vitro or in vivo sources. In addition, drug-drug interactions may be characterized by simulating the co-administration of multiple compounds and their respective impacts on the study drug's absorption, distribution, or metabolism. There are, in fact, many potential applications of PBPK through the entire drug development process. However, exactly how PBPK is applied tends to shift as development programs progress, as the key questions of concern and available data are similarly evolving. For example, one of the earliest applications of PBPK is the selection of the first in human dose. Later in a program, PBPK may be used to characterize drug-drug interactions and food effects, or to extrapolate to special populations, such as pediatric patients or those with organ impairment. As development progresses, the PBPK model that supports it is continuously refined and validated as new information becomes available, a process which is often referred to as the learn and confirm paradigm. In today's talk, we will focus mainly on applications of PBPK in the early preclinical phase of drug development. Before discussing preclinical applications of PBPK in more detail, I would like to say a final word about its application in later development stages. One of the things we are particularly proud of in Simcyp is that our technology has been employed to make over 300 label claims in lieu of clinical trials, to the benefit of over 90 new drug approvals. Most such claims would require the full Simcyp simulator. However, Simcyp Discovery, which I will talk more about later, is forwardly compatible with Simcyp Simulator. Thus, using Simcyp Discovery for PBPK in early development would facilitate a seamless transition towards its application in later development. Now let's talk about some of the challenges of early drug development. There are a number of key questions that typically must be addressed when trying to translate from the preclinical to the clinical phase, such as, what do we need to do to improve the PK of our drug? Which drug candidate should we take forward? What is the projected human dose for our drug candidate? Is it a once-a-day agent? How can I improve absorption? And what is the DDI risk for this compound? PBPK can help to address these questions. Within Certara, we have developed a new tool called Simcyp Discovery, specifically with these types of applications in mind. So what is exactly Simcyp Discovery? It is a new PBPK simulator from Simcyp specifically developed for discovery and translational scientists. It is derived from the gold standard Simcyp simulator and supports PBPK and PD simulations in humans and several animal species. Simulations in Simcyp Discovery generally incorporate information specific to experimental subjects, such as demographic characteristics, organ sizes, blood flows, and enzyme abundance. In addition, compound-specific information, such as phys-chem, protein binding, solubility, permeability, clearance, and transporter involvement may also be incorporated into the simulations. Because the platform supports simulation of both human and animal species, it facilitates translation of PK from animals to humans. Simcyp Discovery also includes a static drug-drug interaction, or DDI calculator, to enable early assessment of DDI risk. Finally, Simcyp Discovery offers a parameter estimation capability, which may be used to fit PBPK models to experimental data. This slide summarizes the four main use cases for PBPK in preclinical development, and by extension, applications of the Simcyp Discovery platform. First in human dose prediction, compound triage and pipeline optimization, DDI screening, and early formulation simulation. Let us first discuss the use of PBPK to select a first-in-human dose. As illustrated in this slide, there are several potential pathways to selecting an FIH dose. Historically, the primary methodology used for FIH dose estimation was allometric scaling, or AS. However, AS has significant limitations in that it is based almost exclusively on in vivo animal data, which can be expensive and time-consuming to generate. Further, predictions based on AS may be significantly biased, especially for drugs that are extensively metabolized or when transporters are involved. Using PBPK, it is often possible to predict in vivo human PK based only on in vitro data. In some instances, such as drugs that exhibit minimal metabolism or whose metabolism is well understood, such predictions may be sufficiently robust to support FIH dose selection in and of themselves. However, more commonly, animal data is used to validate the model predictions, and if needed, refine the model to address any perceived deficiencies in its predictions. Regardless, a PBPK-based FIH dose selection strategy generally requires less in vivo animal data than that of a fully AS-based approach. The next few slides summarize some of the key steps for a hypothetical case study in which animal data are used to validate a PBPK model. In this initial step, we use in vitro information to predict in vivo animal PK. Compound X is a lipophilic neutral compound which has high plasma protein binding across species. The blood to plasma ratio is assumed to be 1. It is cleared via cytochrome P450 metabolism, and microsomal intrinsic clearance values are available across species. Microsomal binding data is also available to correct for binding in these in vitro assays. Solubility is reasonable, given the log of the octanol water partition coefficient log P. Permeability data is not available, so has been predicted based on PhysChem data. Preclinical IV and oral PK data over time are available in both rat and dog. The rat IV and oral PK data are shown in the plots on this slide. The rat IV data was generated at a dose of 1 mg per kg and the oral data at 2 milligrams per kilogram. In this slide, the in vitro-based predictions of the PBPK model are overlaid with in vivo rat data. As you can see, the model predictions indicated by the green lines are in good agreement with the observed rat data, which are plotted as solid circles. The model predictions appear to be accurate for both the IV and oral administration routes. This provides confidence in the model parameters and assumptions. Now that our confidence has been bolstered by validating the PBPK model in animals, we now proceed with predicting PK in humans, for which we have predicted PK over a dose range of 10 milligrams to 1,000 milligrams. As shown here, outputs from Simcyp discovery simulations may include both concentration time plots and derived PK parameters such as AUC and Cmax. One further inference that can be made from this slide is that within this dose range, the exposures are approximately dose proportional, as indicated by the parallel or the approximately even spacing of the profiles in the plot. If you'd like to see a more detailed demonstration of the first in human case study, one is available on demand on our website, the URL for which is provided here and will be available in the recording of this program. Another use case for PBPK in preclinical development is to facilitate compound triaging and pipeline optimization. Simcyp discovery can be used early in a project to define the properties required to obtain a particular product profile. For example, what intrinsic clearance and potency is needed to obtain a particular dose and dosing regimen? The plot on this slide, in which intrinsic clearance is on the x-axis and potency is on the y-axis, helps illustrate this. The different lines represent different dosing regimens. At the beginning of the project, the compounds may not have the optimal properties, but PBPK models can be used to derive heat maps like this to help determine the optimal in vitro properties for a clinical candidate. The Simcyp discovery platform also supports simulation of in vivo PK for multiple candidate compounds as a single batch. The project team may then tabulate, evaluate, and rank the predicted PK and PD performance of the candidate compounds relative to the target product profile. Using simulations based on in vitro data allows a broader set of candidates to be considered than would be feasible if all of these candidates were tested straight away in animals. This, along with a rigorous candidate selection process, helps to ensure that an optimal candidate is selected, thereby maximizing the likelihood of the program's success. Simcyp discovery also includes a static DDI risk calculator. This calculator uses the regulatory guidance equations from the EMA, FDA, and PMDA to calculate DDI risk. These calculations can be done at the perpetrator and victim level for enzymes and transporters, and can be used to guide more robust assessments of DDI risk, such as PBPK modeling or actual clinical pharmacology studies. In this example, the DDI calculator is indicating that there is potential DDI risk associated with both CYP or cytochrome P450 and PGP involvement. So further investigation of the DDI potential of this drug may indeed be warranted. Our final use case for today, we consider the application of PBPK to early formulation development. For example, if absorption is poor, what are the sensitive parameters whereby absorption may be improved? In this slide, a PBPK model was used to evaluate the impact of particle size, solubility, and permeability on the fraction of drug absorbed. As evidenced by the red line in this plot, it seems clear that increasing solubility through spray-dried dispersion has the greatest potential to improve the drug absorption. To recap, why should we use PBPK in early drug development? First of all, it allows integration of all available discovery data to provide an informed PK and dose prediction. It allows scaling from in vitro data with minimal in vivo data, informs decision-making at all stages of discovery, and currently available tools are quick and easy to use. That concludes my presentation today. We want to thank you for joining us, and we'll now open the up for questions.
Excellent. Thanks so much, Mark. It looks like Hannah will be joining us to help out with the questions. So someone wants to know, how do you account for differences in drug metabolism or rate of metabolism in animal species, for example, rat or monkey, when translating PK to humans?
Maybe I can answer this. So I guess when we do this modeling, what we do is chemical validation first. So we build a model in rats using the rat in vitro data. We build a model in human simulation. So we don't necessarily translate metabolism differences directly from rat in vivo to human in vivo. We do it kind of bottom up within each species. So we would understand how the rat metabolism data in vitro, for example, from microsomes or hepatocytes, predict rat in vivo, do the same in dog, and then build in the human. And then depending, I think, on the understanding that we have around the clearance mechanism, may look at alternative approaches.
Hannah, sorry to interrupt. I think your sound, folks are saying it's a little bit hard to hear you, so you may want to make sure you're kind of, like leaning into your microphone. Thank you.
Okay, so what I was saying is that we really, is that better?
That's better, thank you.
Yeah, so what I was saying was that we really try and build confidence within each species around clearance prediction first. And so we try to build confidence with the rat microsome data and rat hepatocyte data predicting rat clearance, for example, rather than scaling directly from in vivo clearance in animals to in vivo clearance in humans. And so we build the confidence from preclinical species, and then we can also look at alternative clearance prediction methods depending on what we understand around the clearance mechanism of a molecule.
Great, thank you. Someone wanted to know, how do you address study-to-study variability in animal studies?
So we can build models for different studies. So if you've got one study that in vivo study that's generated one set of data and another in vivo study that's generated another set of data, then we can incorporate that. Or alternatively, if you're referring more to in vitro variability, then for things like permeability, we recommend that you run positive controls so that we can kind of calibrate the data before it goes into the model, and we can do that with other in vitro data as well.
Excellent. Someone says, thanks for the great talk. Thank you for the kudos. If I have a lead compound or drug, how do I approach it? How do I use Simcyp Discovery to help me evaluate my compound?
So I think the first thing that you would do is you'd want to look at kind of all your different inputs. So the key inputs for the model are things like phys-chem data, protein binding, in vitro clearance or in vivo clearance estimates, and some absorption parameters like permeability and solubility. what you'd do is you'd kind of put those inputs into the model and try and build a model in rat, confirm that you can predict the rat PK. And then if you have any other species PK, do the same, and then run your human PK simulation. So it's all around building confidence with the data that you have. And we would be happy if anybody kind of wants to do this, and obviously use Simcyp Discovery. We'd be happy to kind of walk you through that process in terms of building your model.
Great. Someone wants to know if you can predict rabbit ocular dosing from human data. There, as background, they say we're repositioning a drug that went through phase 1 studies as a tablet, and they're moving to an ocular drop formulation, and they have to do rabbit PK ocular studies.
Yeah, we wouldn't be able to do that in Simcyp Discovery, but we do have our kind of regular Simcyp simulator where we may be able to investigate that.
Okay, fantastic. These have been some great questions. Thank you to our audience. Looks like we've got one last question. Someone wants to know, how can we predict combination studies with more than 2 drugs with these models?
So we can't do that in a time-based manner within the Simcyp Discovery simulator. We do have a static DDI calculator, which would allow you to calculate DDI risk using the equations coming from the regulatory guidance document. But to do a full kind of time-based DDI simulation, you would need to use the full simulator.
Great. Looks like we've got one more question. Can the PBPK modeling software make estimates regarding drug release rates in animal species, which can be later translated to humans?
Yeah, so if you're referring to controlled release absorption, the model is able to model and describe controlled release absorption. So what you may do is you may kind of develop a controlled release tablet that you use in animals based on in vivo studies. We can, and together with the model, you can derive the release rate from the controlled release tablet, and then that could be kind of extrapolated to human and used in the human model. Alternatively, you can kind of generate release rates in vitro, which could then also be extrapolated to human if you had an understanding of your IVIVC. But yeah, that is something that's entirely possible and something we've used it for internally.
Okay, fantastic. Well, thank you so much to Mark and Hannah for that excellent presentation. And I'd like to thank the audience for their fantastic questions. And I'd like to thank AAPS again for hosting us today. And I will pass it back to Stacy from AAPS to wrap it up.
Thank you, Suzanne. It's been a pleasure.
Thank you. It's been a pleasure.
Thank you so much, Suzanne. Thank you, Mark and Hannah. Thank you, everyone at Certara, for partnering with us today for this eChalkTalk. For those of you who are listening to us live, and for those of you who participate on demand, this presentation has been recorded. It will be available in the on-demand section of the AAPS e-learning site. Thank you again, and we look forward to seeing all of you at a future eChalkTalk. You may now disconnect from the webinar.
Thanks. Bye-bye.